Happy New Year to all of you. We've gathered here to listen to a fantastic lecture. This is actually the first in a series of lectures being initiated by the Indian Academy of Sciences. This is the Frontiers of Science lecture, the first lecture in the series that the Academy is initiating. The Frontiers of Science lectures, we hope, will be held in multiple cities, but we start with Bangalore. The Indian Academy of Sciences is headquartered in Bangalore. It was established in 1934 by Sir C.V. Raman, and it was started at the Raman Research Institute. And the major reason why this academy exists is essentially to promote, recognize, and accelerate science. That's what the vision of the academy is, not in these words, but paraphrasing the long set of vision statements of the academy. So today we have gathered to listen to one of these lectures called Frontiers of Science lectures. It will be delivered by Dr. Eric Green. Dr. Green holds an MD and a Ph.D. degree. He is currently the director of the largest human genome institution in the world. It's a National Human Genome Research Institute at the US National Institutes of Health. He's been the director of the institute since late 2009. Prior to that, he was the scientific director and chief of the genome technology branch of the institute. He was earlier the director of the NIH Intramural Sequencing Center as well. Dr. Green received his PhD and MD degrees from Washington University, and he worked under a very famous geneticist by name Maynard Olson. And there he launched his career in genomics research. In 1992, he was appointed as an assistant professor of pathology and genetics and a co-investigator in the Human Genome Center at the Washington University that played a major role in catalyzing the Human Genome Project. Dr. Green was at the forefront of efforts to map, sequence, and understand eukaryotic genomes. Human Genome Project, as I mentioned, was one. He's also very accomplished in the area of comparative genomics, and he's obtained deep insights on genome structure, function, and evolution through comparing genomes of various organisms. He's also identified and characterized many human disease genes, particularly in some forms of hereditary deafness, vascular disease, and inherited peripheral neuropathy. Dr. Green provided leadership to very high-profile genomics efforts, some very high-profile genomics efforts. One of them is the Smithsonian National Human Genome Research Institute exhibition called Genome, Unlocking Life's Code, which is mounted in the Smithsonian Institution, Smithsonian Museum in the US. Science Museum in the US. He's also initiated the NIH program on big data to knowledge, genomic data sharing policy, US precision medicine initiative, etc. He's a fellow of the American Society of Clinical Investigation, Association of American Physicians. He's been awarded with the Kotloff Lectureship Award from the Academy of Clinical Laboratory Physicians and Scientists in 2011. He's received the Wallace Coulter Lectureship Award from the American Association for Clinical Chemistry. He's the founding editor of the journal Genome Research from 1995 when the journal was founded, continues to be an editor of Genome Research. He's a series editor of Genome Analysis, a laboratory manual that many of the students here must have seen and read. He's a co-editor of Annual Review of Genomics and Human Genetics. Dr. Green has authored over 370 scientific publications. Like I said, he is going to deliver the first Frontiers of Science lecture of the Indian Academy of Sciences. And the title of his lecture is Entering the Era of Genomic Medicine, Realities and Opportunities. It's my pleasure to welcome Dr. Eric Green. May I also request you to turn your cell phones off. Thank you.
Well, thank you, Partha, for that very generous introduction. Good afternoon, everyone. Happy New Year. Happy new decade. It is a pleasure to be here. Happy to use that if you want. I am delighted to be giving this inaugural lecture as part of this new series. I'm also delighted to be back in India. I visited here 8.5 years ago, and I learned a lot about what was happening in genomics and data science, bioinformatics, and I was very excited to come back when Partha invited me to come back at this time and get an update. What I thought I would do today, and seeing a lot of students in the room, which I always get excited about, is because it gives me an opportunity to really provide a context for whatever you're studying. I suspect you're studying something related to genomics, or you probably wouldn't be here. And I think understanding a context of what the incredible opportunities you have in front of us by sticking your toe or your knee or your ankle or whole body in this new emerging field of genomics will be, I hope, very stimulating to you for your studies. The theme of my talk, I can tell you now, is going to be how the genetics and genomics ecosystem is rapidly changing, has been rapidly changing, and that the entire ecosystem is one where the opportunities are simply going to be spectacular going forward. And much of what I'm going to describe is almost from a historical point of view, is somewhat of a journey, if you will, a journey of opportunities and a journey of research accomplishments and of some really exciting things that lay in front of us. So what I'm going to go through over the next hour, really cover three major areas. I'm going to talk about the first 3 decades of this still relatively young field of genomics. I'm going to talk about the second part of my talk, sort of the realities and opportunities that we now face at the current stage. And then I'm going to talk a little bit about the future. Now, as you heard, I actually am director of an institute at the National Institutes of Health in the United States. I'm the director of the institute that's responsible for genomics. My main responsibility is to take research funds that we have and disperse them to researchers actually around the world to do research in genomics. I can tell you as a funder, there's sort of 3 things you do. You celebrate, and that's the past. That's the first 3 decades. You worry. That's the second part of my talk, realities and opportunities. And then you plan. And then that's the third part of my talk. So I'm going to talk about celebrating. I'm going to talk about worrying. I'm going to talk about planning. So let me start with the celebrating part. First thing to appreciate, and I hope people do appreciate, that genomics is a relatively young discipline. It is nothing as old as cell biology and biochemistry and physics and chemistry. In fact, the word genomics was first coined in 1987. If you go back in the literature, the word never existed in the biomedical research literature before 1987. It was coined in a lead editorial of a brand new journal that was created called Genomics, where the editors talked about this newly developing discipline of genome mapping and sequencing that included analysis of the information. They've adopted this term genomics. And the reason why this term and this field was basically created in 1987 came from a series of major advances that took place starting in the 1950s with the discovery of the double helical structure of DNA, but in particularly accelerated in the 1970s and the 1980s as molecular biology and DNA cloning came into the fold. We got good at manipulating DNA, cloning DNA, and also sequencing the letters of DNA, such that by the late 1980s, it became clear that we could start to tackle all the DNA of an organism, its genome comprehensively. We could have a discipline that involved the comprehensive study of genomes, and we could think about ways in which we could completely characterize the genome of an organism, such as humans. And of course, what this was really leading to in the late 1980s was the launch of this audacious project called the Human Genome Project, which was launched in 1990. I was very fortunate. I was in the right place at the right time and got to be a participant of the project on day one, on the front line, and participated in it from the beginning until the end. This was a large, international, historically remarkable effort that set out to, for the very first time, read out the roughly 3 billion letters that make up the human genome, and do this in a fashion where all the data would be shared and would be available to anybody with an internet connection anywhere in the world. And 13 years after the beginning of the project was completed, and now we look back at 17 years ago, we sort of mark the end of the first journey of this discipline of genomics, the first journey being the Human Genome Project. But, you know, that was 17 years ago, and genomics has been around 30 years. And so I thought it might be helpful in summarizing the first 3 decades of genomics to sort of give you a highlight reel. In other words, I'll give you 6 highlights from genomics that I think are all worth celebrating. I actually already told you about the first one. And the first one I think will always be number 1 on the chart. We sequenced the human genome for the very first time ever by the Human Genome Project. And that 17 years ago will always mark a major milestone in the history of science. Well, having completed this, I can tell you as somebody who had spent 13 years of their early career in trying to actually read out the human genome sequence, the immediate question was, what's next? In fact, the whole field of genomics said, okay, well, what's next? And I can tell you my institute, National Human Genome Research Institute, asked the question, well, what's next? We have the human blueprint in front of us. What do we want to do with it? Well, it actually was a fairly obvious answer. It was very clear, both to the general public and to the scientific community, that it wasn't just about genomics, but it was about what we were going to do with that genomic information in the long term. Why did we do the Human Genome Project in the first place? And both the popular press in the United States and the scientific press, like Science Magazine, immediately recognized the words that had to be juxtaposed together, genomics and medicine, because it was very clear that there was significant interest in getting the basic blueprint of humans to use it as a tool to improve the practice of medicine. Because it was very clear that the way we had learned to practice medicine was all based on the average patient, and yet no patient is average. Every patient is slightly different, and there are many things that account for their differences, but among those things that account for their differences, a significant amount of it is subtle differences in their blueprint, subtle differences in their genomes, different spellings, that play a role in their health and disease. And if we could apply genomics to medicine, maybe we could improve the practice of medicine by stimulating this field of genomic medicine. However, what I can tell you is 17 years ago, when the Genome Project ended, our view of what genomic medicine was all about was really quite blurry. We could put the words together, but what did that actually mean? Well, we could define it, and we went forth and actually did define it, and we talked about this emerging discipline that involves using genomic information about individual patients as part of their clinical care. But we also recognize that to actually operationalize this, going from just having 3 billion letters of the human genome in front of us was going to require another journey. And in fact, the journey to genomic medicine is really the second journey that I want to tell you about. One that really started with the Human Genome Project being completed, and one that sometime in the future will lead to the realization of genomic medicine. Now, like the Genome Project and like many other journeys, there are going to be many steps involved in this, and we don't even know what all the steps are. I can also assure you this was not going to be accomplished by one researcher, or one laboratory, or one funding agency, or one country. This was going to involve many laboratories, many funders, many researchers, many countries, also recruiting lots of people from many different disciplines to help figure out how do you progress from the most basic information about a human blueprint to actually changing the practice of medicine. I can also tell you it was not going to involve something that's going to happen quick. This was going to take much longer than the Genome Project. So the analogy I make, it wasn't a sprint, it was more of a marathon, with many people from many disciplines, from many countries running together of what will inevitably be many years before we fully realize genomic medicine. But with that as a vision of where we want to go, how did we actually go starting 17 years ago from the starting line of the Genome Project being completed to where we are today? And what I can tell you is one of the things we do at our institute on behalf of the field of genomics is to think about this progression from base pairs to bedside, or if you prefer, from helix to health, and think about how can we develop guides for this journey, this next phase. And we have actually found that it's been very useful to actually convene the scientific community to help us think through what are the steps that are involved in this journey, what are the grand challenges, and how might we knock those down and actually make progress. And so we did that on behalf of the scientific community, and we published these in very high-profile journals, in this case, Nature. We published the first of these in 2003, literally the day the Human Genome Project ended, and it described what was needed next after the Genome Project to inch us towards genomic medicine. And then the good news is we actually made a lot of progress such that by 2011, we ended up having to publish a new one. And in fact, in the 2011 plan in particular, we deliberately put the words genomic medicine in the title because it really was a guide for the next phase that was going to inch us closer and closer to realizing genomic medicine. So with these two guides helping us on this second journey towards genomic medicine, what have been some of the other key advances on this highlight reel? Well, I will tell you that the second highlight is also a spectacular highlight, and one I'll spend a little bit more time on than the other highlights, because the second highlight has changed everything about genomics. And so I want to tell you a little bit about the history, especially for the students in the room. You are now students at a time when generating data is trivial. Getting genomic data is cheap, it is fast, it is trivial. It wasn't always the case. It absolutely was not the case during the Human Genome Project. And in fact, I can tell you that the day the Genome Project ended, when we put out our strategic plan, we actually stated, and I'll show you the quote in a minute, how important it was to reduce the cost of sequencing the human genome. And now, 17 years later, we can say we've been successful. We have reduced the cost of sequencing the human genome by over 1 million-fold. Let me show you how we got there so you don't take it for granted. This was a strategic plan that came out the day the Human Genome Project ended. It had the leadership of the Institute, including my name as authors on this Nature paper. And among the many things we said, the one thing we called out in particular was we said we needed technological leaps that seem so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. And we gave as an example, however audacious it was, the ability to sequence DNA at costs that were lower by 4 to 5 orders of magnitude than the current cost, allowing a human genome to be sequenced for $1,000 US dollars or less. And we picked $1,000 because it seemed like a reasonable clinical test. It was roughly the cost of a CAT scan back then. We said that seemed reasonable. The thing that was a little nutty was that this happened the day the Genome Project ended. We had just finished our first human genome sequence, and the estimated cost of generating that was about $1 billion. And here we were saying, oh, we just need some technological leaps that would somehow knock 6 zeros off of that figure and deliver what was known affectionately as the $1,000 genome. We didn't really have any idea how we were going to do that. We just said it had to be done. We had just finished sequencing the human genome for the first time in factories that looked like this. Instruments that were the size of washing machines or small refrigerators, hundreds of them in multiple countries, running for multiple years, 24 hours a day, every single day of the year, in order to generate that first sequence. And yet we were calling for eliminating these factories and somehow coming up with some science fiction-like new approach for sequencing DNA and making this to the point where it became incredibly inexpensive. And what I can tell you is 17 years later, we actually got it done. It involved us as funders, funding groups in universities and academic settings. It involved the private sector getting involved and venture capital putting money in. It was wonderful public-private relationships that advanced it. But needless to say, it's basically been accomplished. And there's various things you could look at. You could read articles about it. You could follow graphs we put up that show in green the plummeting cost of DNA sequencing when those next generation sequencing instruments became available about 12 and 13 years ago. And our instruments were no longer this big. Our instruments got down to the point where they were about the size of the podium, or now the current instruments are the ones that will actually fit nicely on the podium. And what's remarkable is that it's even getting cheaper. We have crossed that threshold of $1,000, and in fact, we're probably going to continue reducing the cost of sequencing. And it's not just the cost, it's also the speed. You know, that first human genome sequence that cost $1 billion, it also took about 6 to 8 years to generate. That is not a clinical test, that's too long. But nowadays, with the kind of technologies that are available, you can readily generate a human genome sequence for $1,000, but you can do it in a day or two. In some settings, you can do it in less than 24 hours. And the other reason I'm particularly excited about this is even the current technologies that we use will be replaced. There are better ones still coming. We're not seemingly slowing down and getting better methods for sequencing DNA. For those who are interested in the latest technology, a lot of excitement about nanopores you may have read about, ways of threading DNA through small pores and artificial membranes that read out individual bases one by one. The beauty of nanopores is everything gets really, really, really miniaturized, nano scale. And the beauty of it is that you don't need things that sit on podiums. In fact, you can sequence genomes, including human genomes, on devices that fit in your pocket. This is one of the companies, Oxford Nanopore, Oxford Biosystems, and they put out a nanopore instrument. This is the whole laboratory. This is it. It's a lab on a chip. You could sequence DNA reliably on this instrument right now. You can actually start to now sequence human genomes on this. And when I say it's the laboratory, I mean it is the whole laboratory, except you need a laptop, because this is a USB port, so you need a laptop. You can actually use a PC or a Macintosh laptop, so that's pretty good. You're not, so this is remarkable that this technology has improved so much. It also means that DNA sequencing is quite portable. You don't need laboratories. This is this instrument being used in a recent Ebola outbreak in Africa where the infrastructure did not exist, but they could type Ebola viral strains from village to village. This is the same instrument in Antarctica sequencing environmental samples. And this is this instrument being used for the first time in space. They sequence DNA in the International Space Station using this instrument. Oh, and by the way, the same company claims they're coming out with an adapter for your smartphone. You'll be able to sequence DNA on your smartphone, they believe, fairly soon. This is just remarkable. I emphasize this to show how technology has changed everything and is continuing to improve it. It is going to drive advances forward. Well, armed with the ability to sequence genomes inexpensively and fast, what do you think has happened? Well, you know us genomicists, we like our data, and we like sequencing genomes. And so we weren't going to stop with one reference sequence to the human genome. We wanted to sequence many, many people. We wanted to catalog the differences among people. Any two people's genomes have roughly 3 to 5 million single nucleotide differences, about 3 to 5 million polymorphisms. And we wanted to know, and most of those are very common in the population, we wanted catalogs of those. We wanted to have those available to figure out which ones might be functionally important, which ones might be medically relevant. In order to do that, if we just sequence enough people, we can catalog. And so we've gone from having one reference sequence to now having hundreds of thousands of human genome sequences. We're probably going to deal with millions of genomes. In fact, I'll show you data to convince you of that. And with that, we've gone from knowledge of maybe tens of thousands of places across the 3 billion letters of the human genome where we know people vary, to now know of well over 100 million places in the human genome where we know people vary, we know what the variants are, and increasingly our catalogs are becoming richer and richer knowledge about human genomic variation. Of course, to know what the functional consequences of variation is in the genome, you need to understand the function of the human genome. So we need to go from the basic letters, G's, A's, T's, and C's, 3 billion of them in order, to actually knowing where are the genes, where are the regulatory elements, where are the regions that are modified by epigenomic marks, and so forth. And here, we've made quite a bit of progress in 17 years. I think we've made profound advances in understanding how the human genome functions. We now know we have roughly 20,000 genes. We now know that there's far more non-coding regions that are functionally important compared to coding regions. Those 20,000 genes only account for 1.5% of our letters in our genome. Many, many more percent are involved in regulating those genes and doing a lot of complex choreography to give human biology its complexity. It's not in our gene number, it's in the choreography of what we do with those genes. And of course, we've also learned a lot about epigenomic marks on our DNA and how that influences genome function as well. There's a lot that's happening here. I would say we've made substantial progress, but again, for some of you, I hope you dedicate your research careers to characterizing the human genome. We have a long way to go to fully understand all the nuances of genome function, and it will require a lot of new creative ideas to be comprehensive in understanding the complexities of the genome. But we've gotten enough information, especially about genes, coupled with enough information about variants, to now move to the next highlight. And the next highlight really does relate to human disease. We now have made significant advances in unraveling the genomic basis of human disease. Where we've seen the greatest progress has been in rare diseases, diseases that involve mutations in a single gene. Here, this has become remarkably efficient and straightforward, and we've made terrific progress. I'll give you some numbers. So if you think of single gene diseases, these are rare diseases, and there's some well-known ones, Huntington's disease, sickle cell anemia, cystic fibrosis, and thalassemia, and so forth. When the Genome Project started in 1990, there were 61 rare diseases we knew what gene was mutated. 61. Today, there's over 5,000 rare diseases we know what gene is mutated. We've gone from 61 to 5,000. And nowadays, there's just dozens that are discovered every single month. So there's probably about another 2,000 or 3,000 rare diseases we haven't figured out yet, but we're making great strides in figuring those out. So rare diseases, substantial progress. But most human diseases, and certainly healthcare burden worldwide, are not rare disease. It's an aggregate. It's about common diseases. It's diseases like diabetes and Alzheimer's and cardiovascular disease, mental illness, cancer, and so forth. Here, the diseases are not caused by a single gene and a single variant, but rather it's multiple genes, multiple variants, each with smaller amounts of effect that somehow interact and often have a greater contribution of the environment or other factors. But here, there's been great strides. There's been very large association studies done. Those who are sophisticated in this area will hear about genome-wide association studies, or GWASs. This is where you do statistical correlations between hundreds of thousands of genomic variants and then a particular disease, and you find regions of the human genome that statistically associate with getting that disease, but it's only association, it's not a causation. But we've made great progress. We have hundreds of thousands of regions of the human genome now been associated with discrete human diseases. The tough part is now going forward and figuring out specifically what is the variant in that region that is causing that increased risk for disease. And I would just tell you that I think this decade, compared to last decade, this coming decade, will be marked by substantial advances in our understanding the genomic basis of common diseases, whereas I think last decade was much more about understanding the genomic basis of rare diseases. So stay tuned. Hopefully some of you are involved in that. The last highlight brings me great pride because, to be honest with you, I would almost predict, when I was here giving a lecture 8.5 years ago, I doubt I talked about what I'm about to be talking about. Because 8.5 years ago, we had almost nothing to say about the actual practice of medicine. But I come back here 8.5 years later, and I can say that we now have vivid examples of genomic medicine emerging. It's no longer just an idea. We're actually starting to see genomic medicine in action. We've gone, since the end of the Human Genome Project until now, from having a very blurry view about what genomic medicine is all about to actually having some idea of what it might be about. It is starting to come into focus. It's just some of the earliest examples, but I think these examples are highly illustrative. I don't have time to describe them in detail, but just if you want to watch on your radar screen, cancer is absolutely now being significantly changed how we manage and diagnose cancer based on genomic information. Cancer is a disease of the genome, and number one application of genomics in medicine is already cancer and going to continue to be cancer going forward. A second thing is rare diseases. It is now routine for when a patient is on a diagnostic odyssey, you don't know what is wrong with that patient, they seem to have a rare disorder, you just get their genome sequence. Over 50% of the time, you will figure out what is wrong with them. It is now mainstream. Hundreds and hundreds of people every month around the world are getting their genome sequenced as part of a diagnostic workup for a rare disease. Pharmacogenomics is coming into the fore. Why is it we all respond differently to medications? A big part of it is differences in our genome that influence drug metabolism pathways. We are discovering the specific variants that influence those pathways. That's becoming a testable, a clinical testable situation to help physicians decide which among known medications they should give an individual patient. The fourth area I always like to point out is prenatal genetic testing, which can now be done by non-invasive methods, detecting small amounts of fetal DNA that float around in the maternal bloodstream. And we are now seeing, that is the number one genomic medicine test worldwide. Upwards of 7 to 8 million pregnant women will get non-invasive prenatal genetic testing done this year around the world. That is a remarkable thing. It didn't even exist 8 years ago, and now it's the number one genomic medicine test. So bottom line, genomic medicine is here and now. What I just described to you, these 4 examples, are just the tip of the iceberg. There's going to be a lot more. I can guarantee you this decade is going to see many examples of additional implementations of genomics in medicine. And there's a lot of more excitement to come, although it's still not straightforward. It still requires a lot of hard work. And in fact, this is actually a good place for me to transition to the second part of my talk. The second part of my talk is to shift gears. If you haven't noticed so far, I can be a little enthusiastic about genomics. I admit it. And so I like to sort of balance that by talking about realities. And the realities are, I don't want you to think that all this is simple, because we face currently some big realities, and this is the challenges that we are now facing in genomics. Of course, with these challenges come really good opportunities. So I'm going to tell you about 6 realities that really temper my enthusiasm, but each case I'll give you optimism about the research opportunities that they give you. So, reality number 1, I already alluded to it. Generating a human genome sequence, reading out all the G's, A's, T's, and C's of a person's genome is trivial. It is straightforward, it is inexpensive, it's relatively fast, it can be done in all sorts of settings. Getting that data is straightforward. If you get that data on a patient, though, you then go and you want to round on that patient the next day. And even if you're handed a report that gives the 3 to 5 million genomic variants that that patient has compared to a reference genome, I can guarantee you that almost all of those genomic variants on that list, you will have absolutely no idea what it means. And that is because there is a significant gap between our ability to get the data on a patient and our ability to interpret that data. And it's not that the literature is not filled with lots and lots and lots of papers that are reporting information about variants and their relative importance for medicine. It's just that the busy physicians who are taking care of all these patients simply don't have time to assimilate all of that information. There's a gap in our ability to synthesize the data on genomic variants and understand what it means clinically. So getting a clinically understandable view of a patient's genome is not trivial, and we need to close that gap. There are major research efforts ongoing, some by us, some by other countries, I can tell you, to try to develop resources for busy healthcare professionals to be able to quickly look up every variant in their patient and be able to know which one is clinically relevant, which one is not. But don't think just because you can get the data, it means can you interpret the data. It's not the case. This is a huge challenge that exists right now and one that we are trying to solve. Second reality. The field of genomics has changed radically, even in 17 years. And that also brings some big challenges. What do I mean by that? Well, during the Human Genome Project, I can tell you that up until about 17 years ago, genomics was only about biomedical researchers. It was just about nerdy scientists like me at the bench, mapping and sequencing DNA. People at computer terminals analyzing the data. It was just researchers. It was a very closed group. 17 years ago, when we started to think about genomic medicine, we were able to grow our field. I think healthcare professionals started to come into our tent and recognize that genomics might have some relevance for the practice of medicine. But still, it was just biomedical researchers and healthcare professionals involved in genomics. But as genomic medicine has become a reality, all of a sudden, for cancer patients, for patients with rare diseases, for their families, for patients who are getting pharmacogenomic testing, for pregnant couples who are getting non-invasive genetic testing, all of a sudden genomics is becoming part of the language of their medical care, which means all of a sudden genomics becomes relevant to patients and their friends and their families, which means genomics becomes relevant for society. So once upon a time, we didn't have to think about much except for figuring out how the genome was put together and figuring out how we might implement genomic medicine. But now we have to think about societal implications of genomics, because now we have touched something very complicated, and that's healthcare. And the second you touch healthcare, you are dealing with a lot of additional complexities. You're dealing with how it gets paid for, if it gets paid for. You're dealing with how it gets regulated, if it gets regulated. You're dealing with making sure healthcare professionals, as well as patients, understand the language of genomics enough to understand what they're each trying to communicate to one another. And you're thinking about things related to privacy as you get genomic information about patients. So we think a lot about this. We even study the ethical, legal, and social implications of genomic advances. But again, this is a big reality, and it's only gotten more complicated, not less complicated, in the last 17 years, because we're starting to touch healthcare, and that brings in a lot of complexities as well. New reality number 3. This is a problem. We created this problem, and we need to solve this problem. I will be blunt. There is a lack of diversity in genomics. This sort of came to the fray in about 2009 for the first time, when some of these big studies that were being done in human genetics, these genome-wide association studies, which involve studying tens of thousands of people and trying to map regions of the genome that contain variants that confer risks for common diseases. When those data were looked at, it became very clear that overwhelming numbers, percentages, well over 95% of the people in those studies were of European descent. They were all basically whites of European descent. So alarms went off in 2009. And actually, the same authors came back 7 years later and asked the question, were we doing any better? And the answer was we were only doing a teeny bit better. And if you want to look at the data, in 2009, it was 96%. By 2016, European ancestry of participants in these studies had dropped to 81%. So maybe that sounds like it's an improvement, but actually when you unpack it a little bit, you realize that there's a 5-fold increase in non-European samples in these publicly available data sets. The problem is that about 78% of the increase came from populations of Asian ancestry. That may sound good until you realize almost all of that was from China. So it wasn't even a very, very diverse representation of Asia. And in fact, many groups, including groups of other ancestral populations, made up a very small percent. And in fact, a more recent cataloging of this, which took place in last year, where the good news is that more and more individuals are participating in these genetic studies. And by numbers, you can see that here. And so the numbers of people for which data is available for scientists to study is going through the roof. The problem is it's overwhelmingly people of European ancestry, and you can see the breakdown as you move, Eastern Asian being the next largest group, and so on and so forth going down. And here you can see it as a percentage. This is a problem for a couple of reasons. One, it's a sociological problem that we are not capturing the world's diversity of genomic information. We must do that. It's actually a scientific reason why you want to do this, is because a lot of things you need to do that when you go to do the data analysis, you need to use as control groups people from the appropriate similar ancestral background. And if you don't have that data, you don't have the comparative group to study, and therefore you lose the power of your analysis. So scientific reasons and social reasons why this must be fixed. I can tell you our institute is committed to fixing this. If you're interested in how we're doing that, we last year wrote an article, or 2 years ago in Nature Reviews, Genetics about this, and I encourage you to look at that. But we are now overemphasizing the acquisition of samples from individuals of non-European, who are not of European descent, in order to help sort of fix the deficiencies we have in the populations. Meanwhile, we are strongly encouraging other countries, other funders to get involved and help us, because the worldwide collection of data will be better if all of the data that is available and shared is more diverse. I was delighted to see this, and I applaud the efforts. Partha was a major leader of this. I think others here in the audience were also major leaders of this recent article that was published in Nature that talked about this new genomics initiative coming from Asia and generating what will be a groundswell of new data from diverse, different populations across Asia. And this is exactly what is needed, and we hope we're going to see similar things from other countries as well. Next opportunity, a new reality. There are some major changes that are going on that really I think are worth highlighting, that relates to how genomic data is being generated. And it really is sort of going to be a shift from the more traditional way that human genomic sequence data would be generated by research. And it's going to soon be not being generated as part of research, but rather is going to be generated as part of healthcare. So if you just look at, and there's an international organization, the Global Alliance for Genomics and Health, that has been studying this and trying to predict towards the future. And here is the trend. If you go back a handful of years or even just a couple of years, a minority of the human genome sequence data being generated was generated as part of healthcare. Within 2 years from now, by 2022, it's predicted it's going to be over 80%. Now, this is remarkable when you think about it. I can tell you as a funder, it makes me a little bit nervous because it means that I'm not controlling how genomic data is being generated. Most of it's going to be generated as part of healthcare. But their predictions, which are sort of shown here on this trajectory, much of it coming from sequencing of patients with cancer and of rare diseases, points to the fact that by 2022, it is estimated there will be 40 to 50 million human genome sequences that will be generated. And one of the architects of this study was Ewan Birney, and it was interesting how he has really been stressing in some of his recent writings that the ongoing adoption of genomic medicine means that some of the most fundamental data types of molecular biology, that is genomic sequencing and other genomic assays, are being paid for and organized at scale by healthcare. And what really, that has a number of implications, not the least of which is thinking about what can we do to make sure that we have access to those data for research. And in fact, that's what the Global Alliance for Genomics and Health is trying to do, to figure out ways to make sure we can share this data among countries from different healthcare systems and so forth. That's going to be critically important. And I can tell you it's something that we are keeping a close eye on, again, because of the shift from having research funders dictating how the data is being generated to now in the future where healthcare providers will be the ones dictating how this data is generated, and we want to make sure it is generated correctly. There's another implication here, though. This is the kind of thing that I do worry about among my list of worries, and that is that diverse communities around the world inherently face barriers to accessing genomic medicine. That's just a reality. It's like any new technology in medicine. Diverse communities often encounter barriers. But of course, if most of genomic sequencing is going to be done as part of healthcare, it means we may risk exacerbating the diversity problem I just told you about. Because it could end up being that the great majority of genomic data will be generated from wealthy patients who could afford to get it done as part of the way they get healthcare, and that would be a bad thing. So again, there are implications for all these things, even at a sociological level. New reality number 5. This is a fun one. This is as much an opportunity as anything else. Up until now, you might have thought from my talk that I was conveying the idea that I thought human disease was all about genomics and that was all that mattered. But I know that's not the case. I know that for most human diseases, there is both a genetic component and an environmental social component. I'm a genomicist, so I basically have spent my professional career thinking about this half of the pie chart. And I'm a lucky genomicist because I have been benefited by remarkable new technologies for sequencing DNA and for characterizing this part of the pie chart. But the world's changing because new technologies are becoming available that are getting better and better at measuring our physiology, measuring our environmental exposures, measuring our lifestyles. And that's going to empower scientists to collect a lot of data about people that will help us fill in this part of the pie chart. I'm referring to all sorts of new wearables and mobile health detectors that measure analytes in real time and measure cardiovascular function in real time and do all sorts of things. And the body electric is becoming a reality. The crude version of it are iPhones that do various things or Fitbits that do various things. But they're much more sophisticated research-grade tools that are becoming available. And we're going to collect a lot of data on people using these kinds of technologies. This has led to the notion of not just thinking about genomic medicine, but thinking about precision medicine. And precision medicine is just sort of zooming out a little bit and thinking about not just individuality around genomics, but around other things as well. It's just a more precise accounting for individual variability. I emphasize genomic medicine because it was about individual variability in a person's genome. But as we have these new technologies for measuring lifestyle and environmental exposure and physiology, we will empower scientists to collect data about physiology and collect data about lifestyle and empower them to collect environmental exposures as well as things that are somewhere between lifestyle and environment, like what we eat or what destructive things we do to our body. And all of a sudden, you could have a lot of data about individual variability in multiple different domains. And if you could use such information for the practice of medicine, you could do it more precisely. And so this has led to the notion of combining these various data sets, genomic data, using these two technologies for physiologic and lifestyle data, and for where there are robust systems for capturing phenotypic information acquired through medical care, electronic health record data, which I can certainly tell you in the United States is pervasive now in almost all healthcare delivery systems. And while most of it was created for billing, it's all going to be increasingly available for research. And these areas become very rich data sets. For those of you who are data scientists, go crazy, folks. There's going to be a remarkable future for you when all these data become available. Recognizing that opportunity, various countries have jumped into the idea of doing very large-scale precision medicine studies. In the United States, the big program we launched several years ago was something called the All of Us Research Program. This is a program where we are enrolling 1 million people in the United States to volunteer. When these people volunteer, they are going to participate, and they're going to share their genomic data, their lifestyle information, biological samples. They're going to let us access their electronic health records. They're going to wear mobile health devices. They're going to be followed for probably 10, 20, 30 years. And all that data is going to be available to any researcher around the world, as long as they get permission to access the data and do appropriate things with it. All the while, we'll make sure to try to build in privacy protections here. But the idea is to generate one of the largest precision medicine data resources in the world, but knowing that we'll be doing it with many other countries who are doing this as well. So this is the United States' effort, but we're not alone. In fact, we're not even ahead. I can tell you countries like the UK, through the UK Biobank or Genomics England, are far ahead of the United States or most other countries. But a number of cohort studies, and these are cohorts of people who are being studied, are popping up. In fact, there's so many of them that we just created a new international organization called the International 100K for 100,000 participants or more Cohorts Consortium. These are all the countries that are involved. I do note India is among them, and there's various demographics about what exactly they're doing. But from the earliest cataloging, that's probably now about a year old, there are over 30 such studies that are now on track to capture information, precision medicine information, on well over 60 million participants, and those numbers are going to certainly grow with time. We certainly hope we fill in more of these countries. This will help fix some of the diversity problem as more and more of these, if more and more of this, the world, all the different countries start to fill in with the color, and we hope that will happen. But most importantly, we should recognize that to maximally leverage the value of this data, it would be to share it across all these different countries and get the data sets as large as possible to tease out as much meaningful information out of it as we can. And that's the goal of the IHCC, is to figure out how to share the data. So the opportunity here is we're going to go way beyond knowledge of our G's, A's, T's, and C's and their influence on health and disease. We're going to move into lots of other information from electronic health records, from wearable devices, from lifestyle, from environmental exposures, and so on and so forth. Last, new reality and opportunity. And this is one I specifically am calling out, visiting this country, to really encourage you to think about this. I will just tell you that genomics and precision medicine requires global collaborations. This has absolutely been the case since the beginning of the field. The historic examples I would cite are the Human Genome Project, an international effort. The International HapMap Project, which got a lot of information about human genomic variation, international project. 1,000 Genomes, international project. And the International Cancer Genome Consortium. There are others. These are just 4 I just chose to highlight. In the past relatively few years, there's a growing number of these international consortium because the time is ripe to accelerate and expand this global network of collaborations, because it's all about getting the data, sharing the data, analyzing the data, comparing the data. There are examples of this. I've told you about the Global Alliance for Genomics and Health. I didn't tell you. There's one just around genomic medicine implementation, the Global Genomic Medicine Collaborative. There's one around rare diseases. There's one around common diseases. I told you about the cohorts one. And another one that I'm involved in is something called the Global Biodata Coalition. This is an attempt to make sure that all the data resources that you regularly use, like GenBank and PubMed and various other, Uniprot and so on and so forth, that these are in strong financial footing, that these are available to anybody worldwide, and we really take care of these fundamental data resources that everybody relies on. So that's a coalition of funders to take care of it. Please participate in these. Make sure your country is represented in these. This is how I think a lot of science is going to be done at a global scale, is through highly coordinated international efforts like these. So in the last 5 minutes, I just want to talk about the future and just tell you that we think a lot about what's going to happen in genomics in the coming decade. And we think about it very much with the lens around the fact that the ecosystem has changed so much over the last 17 years since the end of the Human Genome Project, and there's every reason to believe the ecosystem is going to continue to change substantially. Everything I've described to you for the first 32, 33 years of genomics may have sounded like it was straightforward, it was linear, we knew it was going to happen. No. It wasn't simple, it wasn't straightforward. It actually looked much more like this than a straight line. That's just the nature of this kind of science, and that's fine. But what it does mean is with all the twists and turns, it means every once in a while you got to sort of take stock of where you are, and you got to be strategic, and you got to articulate a strategic vision going forward. I alluded to 2 strategic plans. Those strategic plans actually were built on the successes of 3 strategic plans that got us through the Human Genome Project. But then there were these 2 I told you about earlier from 2003 and 2011 that served us well. But the truth of the matter is, even the 2011 strategic plan, which is the one that we are currently still operating on, it's 10 years old now at this point, and it needed to be refreshed. And so we recognized it was time to refresh it, and we wanted to put out something in early 2020, or early in the decade, that would get us what we want. We knew it would be important to us as a funder to make priority decisions. We also knew it would be very valuable for the community. And so this is sort of the third journey. It's the journey going forward and what the next decade is going to bring us. We have a very organized way of doing this. I don't want to walk you through the whole timeline, but we started this. We launched it in 2018. And our plan is to publish our schedule is to publish the new strategic plan next October, or this October of 2020. It will be a commemorative month. It'll be the 30th anniversary of the launch of the Human Genome Project. And in fact, so we've had a number of planning events over the last couple of years. We're sitting here right there, busily putting together a draft manuscript, which we will have available by the spring to get community input and then write a final plan that'll be published in October. So I would just tell you that this is something to look for and to really emphasize how different this round of strategic planning was. It was very different than the one we did in 2003 or the one we did in 2011. And the reason for that is once upon a time, genomics was very limited in who was doing it, and now everybody does genomics. I could put a real figure in front of you to convince you of this. So, very simple. The National Institutes of Health in the United States has 27 institutes and centers. We are one of them, genomics. Once upon a time, if you just looked at what fraction of human genomics research was funded by our institute versus all the rest of NIH. During the Human Genome Project, we funded 95% of human genomics research. Well, yeah, because we were the only ones doing genomics. Greater than 95%. When the 2010, when we wrote the 2011 strategic plan, that had dropped to 50%. We were funding 50% of human genomics research, but other institutes were funding 50% as well. Today, as we are writing our new strategic plan, I can tell you that we only fund 10% of human genomics research. So here we are, where once upon a time when we spoke for all of genomics, because we essentially were all of genomics. Now, when I speak, I can only speak for 10% of genomics, because I only fund 10% of genomics at NIH. What that means is we just need to sharpen sort of our perspective on what we are representing. We no longer speak for all of genomics. It's everywhere. It's into cancer work, it's into microbiology, it's into environmental science, it's all over the place. But what we can speak for, and this will be the main theme of our strategic plan that I hope all of you will read when it comes out in October, is we can talk about the forefront of genomics. So we view our description as the cutting edge, the very front of the spear. We're going to represent the strategic vision at the forefront of genomics, that when we advance the forefront, everybody follows, everybody benefits, and everybody will be able to use genomics for the science that they want to do. So that's the theme that you'll read about in October. If you want to learn more about our planning process, there's a whole website. You can communicate with us. You can follow us on social media. But most important, what I hope you do is when it comes out in October, especially the students, please read the document. It'll come out. I can't tell you what journal, but it might be the same journal that we published the last 2 in. But they got to accept the paper, but they will. So just, you could imagine where to find it, and we will publicize it when it comes out. But please read it, because I really want you to see this as the guide for your personal journey as you pursue research that you're interested in doing. And last thing, if you've enjoyed this at all and you want to stay connected to my institute or to the field or to the forefront of genomics as we describe it, if you want an extra email a month, and you don't have to sign up, but my staff and I put together a monthly newsletter which we send out the first week of every month. that summarizes advances in the areas. A lot of stuff that I talked about will be updated. So feel free to subscribe to that. And at the 10th anniversary of me being director of the Institute, I let my communications director convince me that I had to get into the Twitterverse. So I now have a Twitter account. Anybody who wants to follow me, I'm told that you're supposed to write down the @NHGRI_director and feel free to become one of my followers, and we're actively tweeting all the time now. And that's another way you can stay connected. And for those who don't know what Twitter is, never mind. Ask somebody in the back 7 rows and they'll tell you. So with that, I will stop, and I'm happy to take any questions if there's time. Thank you very much. Yes, that's right.
So, with all this technology that has come from the past and the current one, if you talk about, let's say, the near future, how much of a push do you think machine learning will give to this whole field of genomics?
So I think that the question was, what's going to be the role of machine learning, and maybe we could even expand it to artificial intelligence, to sort of genomics. And actually, I might even generalize your question. I hear a lot of excitement, and I don't have the expertise. There are probably people in this audience that have order of magnitude more, maybe 2 orders of magnitude more expertise than I do in artificial intelligence. But they particularly get very excited about some of the stuff around precision medicine because it's multimodal data. It's, there's genomic data, but then it's going to be interfaced with exposure data and electronic health record data. And so the power of artificial intelligence would be dealing with very disparate data sets, but somehow coming up with patterns that would allow you to interpret things. So it is our belief that that is very important. I can tell you when we talk to expert data scientists who come and talk to us specifically around genomics, they certainly say this is an area where we should be investing, is the interface of artificial intelligence and genomics and precision medicine. So we will be doing that. But admittedly, I think there's great potential, and I think one of the things we really need to do is to sort of better focus in specific areas. We probably need some very exploratory work at first to see really where to drill down.
Keep on going. We really enjoyed your talk, but I have one question. I just said that it's becoming prohibitive to access
it is prohibitive to access the data? Oh, the medicine. Yes. So the question is, the question is, what is the circumstance where discoveries are made, especially around rare genetic diseases, where then when they go to, they figure out a therapy, for example, a drug therapy, and then the only way the private sector or whoever the company can recoup their investment to develop that drug is to make it very expensive. So when I had the thing about new realities, and I had the thing around genomics and society, and I said, and then we have to touch the healthcare system, that's among the huge challenges, is someone's got to pay for this, and someone has to deal with the complexity of setting prices for drugs. By the way, it's not unique to genomics. I can't speak to drug pricing in India. I can speak to the United States, and it's a mess in the United States. But it's not a mess just for genomic-based drugs. It's a mess for almost all drugs. It's just they're really obscene for some of these drugs for rare diseases because when you finally have a company develop a drug for 100 people in the world, you can imagine the price tag is going to be high. So we need to figure out as a society, it's probably multiple societies because different countries, how are we going to tackle that and who's going to pay for it and so on and so forth. It will be, and I think it underlined your question, it will be a tragedy if we get the knowledge and we figure out how to do it and we can't operationalize it because we can't afford it. That becomes a societal decision of how do we afford it. And at the same time, companies people have to be accountable for how they're spending their money too. So it's a very difficult situation. One of the things we're doing, I can tell you in the United States, is figuring out ways to try to de-risk the drug development pipeline, to try to do things where they don't have to put too much money in up front. We try to get or also try to make the process of going from a target to actually having a drug be more efficient, less expensive, so that more academics could get involved in developing new therapeutics, not just all the private sectors. But these are all just experimental ideas.
Yeah, excellent talk. Would you like to
Sure. So the question was, I do give short shrift in this version of the talk to epigenetics. But again, I hope everybody realizes that one of the other things we were aware of before, well, even before the Genome Project, but particularly over the last 17 years, we've become well aware of the fact that the genome function is not all about the order of G's, A's, T's, and C's in our genome, but that the ways our genome is modified through methylation and histone associations and other epigenomic marks have tremendous influence on genome function, and importantly are one of the ways that the environment influences genome function is through influencing epigenomic marks. We also know that some disease is basically caused by aberrant epigenomic marks on our DNA, so it's not always about DNA polymorphisms. And the good news is that you can adapt the methods for sequencing DNA to be able to characterize these epigenomic marks on large scale. And so, among the data sets, and I probably should have said, among the data sets, not only just catalogs of genomic variation, also catalogs of differences in epigenomic marks at different tissues, different stages, et cetera, et cetera. So all of that is absolutely in the mix and will continue to be in the mix because it's clearly a very big, important part of genome function. But the good news is that the tools for sequencing DNA can be readily adapted so that it becomes very inexpensive to generate a lot of epigenomic data. Yeah?
This is a continuation of the previous question.
Yeah.
But the world is a very unequal place. And are these technologies going to enhance that inequality rather than trying to?
Yeah, so I think the question was, are genomic the world is an inequitable place. Absolutely true. Even within a country, huge inequities. And it's why I spoke to this issue around worrying about, all of a sudden, it's really, again, it's this point I want to make. When I fund a study, I could dictate this is the diversity level I want and pay. When all of a sudden I lose that control in healthcare systems, I worry that all my efforts are going to go for naught because now it's going to be only the wealthy who are going to get healthcare and only the wealthy are going to get genomic medicine and so forth. I think your question was even broader than that, just related to genomic technologies and access to this. And I think this is a real issue. I don't think genomics is unique, so any medical advance has applied in the same arena. I think what we have to offer is continuing to push on the accelerator to make things cheaper and cheaper. Because if we can get them cheap enough, then at least we can remove that barrier. There'll be other barriers. It's not always just about money. There's also literacy barriers, and we'll also work on those. So all we can do is to try to learn the best practices of elsewhere. What have been the real success stories? And it's mixed. But what have been the real success stories of new medical advances and having them be distributed in an equitable and fair fashion and not exacerbated health disparities? And can we simulate those the best of our abilities? I think the one thing I think that we have going on our side is the fact that genomics from the beginning has embraced the study, instead of just saying we want to pay attention to the ethical, legal, and social implications of genomic advances, not only are we paying attention, we're funding it, research. And that's been very helpful up until now, because we dedicate 5% of all of our research goes to these ethical, legal, social implication studies, is that when you come to a situation where you need to develop a policy, you need to take an approach, you at least have some data to sort of define the problem in a better way. And so I think we're developing a better intellectual framework for trying to address these things, and that has not always been the case. Oh, yeah. There's a microphone, and then everyone can hear you.
Would you say that the treatment for any disease is moving towards a phase that will work at an individual level, keeping in mind the psychological, physiological, and the genomic factors? Will the treatment be at that level that we'll have to tailor it according to the individual? I
want to be clear. I think we want to do is we want to tailor the care. It doesn't mean design a new medication. I think where we want to be is in a situation where we have as much information in front of us as physicians and other healthcare professionals to tailor the care for that patient. That would be precision medicine. In the case of genomic information, well, we get genomic information. Maybe we'll be in a day where, and we're already, again, some of these mobile health devices are absolutely in use in some settings where you get unique physiological information about somebody, where you're measuring their blood pressure not once when they're in the clinic, but hundreds and hundreds of times and making decisions. Maybe another individual aspect might be better information collected about people's lifestyle, about what they're eating, what they've been exposed to, maybe sign more about their social environment and so forth. So I would say it would be all of those data sets would ultimately be in play for tailoring the care.
One last question to here. Coming few years, it will be more regularly used in clinic. Almost every patient will be checked for their genomic data. So will it lead to discrimination?
Yeah. So the question is, if we really are heading to a world where the vast majority of people have genomic information generated by this product, will it lead to discrimination? And the short answer is, I hope not. And we need to think about how to prevent that. And that's going to have to take place within the legal framework of each country. I don't think whatever solution would work in one will work in another. So for example, in the United States, we have a law called the Genetic Information Non-Discrimination Act that is a law that has so far held up that says you cannot discriminate by health insurance or by employment based on genomic information about a patient. Other countries have passed similar laws. And over time, we need to figure out how to provide sufficient privacy for people's genomic information so that it's not used against them. And that has to require regulatory, legal framework, regulatory framework. It also has to involve sort of security framework to make sure that data is not accessible. I will also say, on the other hand, I don't think genomic information is powerful, but it's not 100% unique. I mean, there is a lot of healthcare information that people have in their medical records, and they could be, in principle, discriminated based on what medications they take or what other family members, what disease. So we should recognize that genomic information is new information, but family history information has been around for a long time. And other healthcare information has been around a long time. And people have been discriminated in different ways in different countries, and that has been dealt with in different ways. So we need to be carefully monitoring it, and every country has to tackle this.
Thank you very much, Eric. This was just awesome. May I request a predecessor of mine, Professor Dipankar Chatterjee, to honor our speaker this afternoon. No, you pass it on. I'll come this way.
Okay.
Lovely talk.
Thank you.
Privilege that you have visited us.
Thank you. Thank you very much.
I also have another announcement. For those of you who can travel 1,500 miles within the next 12 hours, Professor Eric Green is giving the Obaid Siddiqui Memorial Oration in the National Institute of Biomedical Genomics in Kalyani. We would love to see you there. Thank you very much.